What's good, y'all? We're going to now react to the Science News Podcast, episode 4. It's out now on YouTube. So if you haven't already, go check that out. Link in the description below. And if you have, well, let's talk about it. So I'm going to get it playing right now, and we'll just do a live reaction right now.
The interesting part, they're seen as perfectly precise and the more complex reality on the ground, especially with sickle cell.
Yeah, it makes you really think about what fixing a genetic issue actually means, especially the tools we have right now.
Exactly, and that's really what we want to dig into today.
So we've got this really interesting study from Blood Advances. It details a CRISPR method for tackling sickle cell and beta thalassemia, but maybe not in the way you'd first think.
Right, it's not about directly fixing the faulty gene. It's focused on boosting something called fetal hemoglobin.
And to add some perspective, we also found a couple of, honestly, quite sharp opinion pieces that directly question this whole precision angle for these kinds of treatments.
It's so important, isn't it, to be excited by the science, of course, but also to think carefully about the words we use. You know, are we really describing what's happening accurately, or is the hype maybe simplifying things a bit too much?
So that's our goal for you today. Let's unpack the science, look at the potential, but also understand this debate about the language itself.
Sounds good.
Okay, let's dive into the science first. Using that Blood Advances paper, maybe just a quick refresher, what's the core problem in sickle cell and beta thalassemia?
Sure. So in both conditions, the issue stems from mutations in a gene called HBB. This gene codes for a part of our adult hemoglobin.
Right.
In sickle cell, the mutation makes red blood cells distort into that sickle shape, causing blockages, pain, all sorts of problems. In beta thalassemia, it usually means you just don't make enough functional adult hemoglobin. Both are serious.
Okay, so the adult hemoglobin is the problem. What's the therapeutic idea in this study then?
Well, the strategy here is pretty clever, actually. It's about encouraging the body to make more fetal hemoglobin, HbF.
Fetal hemoglobin, like the kind baby.
Exactly. We all have it before and shortly after birth. It's slightly different, and critically, it's not affected by the sickle cell mutation. So the idea is, if you can boost HbF levels in adults, it can compensate for the faulty adult hemoglobin.
So it's like turning back on an old system that usually gets switched off.
Precisely. It's a workaround.
Okay, intriguing. So how does CRISPR make this happen? How does it boost the HbF?
So the researchers didn't go after the mutated HBB gene itself. Instead, they targeted the control regions. Think of them like switches or dials for the genes that make fetal hemoglobin, HBG1 and HBG2.
Oh.
They used CRISPR-Cas9 specifically to disrupt a binding site for a protein called BCL11A, right in the promoter area of those fetal genes.
BCL11A. That name keeps popping up in this field. What's its role again?
Yeah, it's a key player. BCL11A normally acts like a silencer or a brake on fetal hemoglobin production after birth. It helps manage that switch from fetal to adult hemoglobin.
So by disrupting its binding site?
By disrupting where BCL11A normally latches on, the researchers are basically taking the foot off the brake. They're allowing those fetal hemoglobin genes, HBG1 and HBG2, to become active again in adult cells.
Got it. So not fixing the original typo, but rewriting the instructions for a different gene to compensate. How do they actually get the CRISPR machinery into the right cells?
They used a method called electroporation. Basically, they give the cells a little electrical zack to make temporary holes in the membrane, allowing the CRISPR-Cas9 complex, that's the enzyme in its guide RNA, to get inside.
And which cells are they targeting?
They're targeting the CD30 plus hematopoietic stem and progenitor cells, HSPCs. These are the really important ones, the stem cells in your bone marrow that give rise to all your blood cells, including red blood cells.
Okay, so edit the source and the effect should persist.
That's the goal. If you edit these stem cells, then all the red blood cells they produce down the line should carry that edit and hopefully produce more fetal hemoglobin.
Makes sense. So what did they find when they tested this in the lab, in vitro?
The lab results looked very promising. They achieved high rates of the desired edits, the indels, right at that BCL11A binding site.
And crucially, did that lead to more fetal hemoglobin?
Yes, significantly more. When they let these edited HSPCs develop into red blood cell precursors, these erythroid progeny showed a clear increase in HbF production.
Any downsides at that stage? Did it affect how the cells grew or matured?
That was another important check. They found that the editing didn't seem to mess with the cell's ability to mature properly or to undergo a nucleation, which is where they eject their nucleus to become functional red blood cells. So that looked good.
Okay, promising start. What about when they moved it into animal models, the in vivo experiments?
Also positive. They transplanted these edited human HSPCs into mice. The cells engrafted successfully. meaning they settled into the bone marrow and started producing blood cells.
And did it disrupt the normal balance of blood cells?
Apparently not. They checked the different lineages, T cells, B cells, myeloid cells, red blood cells, and the development seemed unaffected by the presence of the edited cells. Plus, the gene edits themselves, the indels, were found to be stable and persistent in the bone marrow over time.
Okay, that's encouraging. And the key question, did they see more fetal hemoglobin in the mice too?
They did. The red blood cells derived from those edited human stem cells in the mice showed increased HbF levels.
And did they connect that increase to an actual functional benefit for sickle cell?
Yes, and this is a really key finding. They took HSPCs from actual sickle cell disease donors, edited them using this method, let them develop into red blood cells, and then exposed those cells to low oxygen.
Which normally triggers sickling.
Exactly. And they observed a significant reduction in red blood cell sickling in the treated cells compared to untreated ones. That's a direct demonstration that the increased HbF achieved through this editing can combat the disease phenotype, at least in this model.
Wow, okay, that's quite compelling. But of course, with gene editing, safety is always the big question. What about side effects or unintended genetic changes?
They definitely looked into this. One thing they observed was a specific larger deletion, about 4.9 kilobases occurring between the two fetal hemoglobin genes, HPG1 and HPG2. This happened at a noticeable frequency initially.
Is that deletion a problem?
Well, they investigated further by looking at individual cell clones. Their analysis suggested that even if one copy of the HPG2 gene was lost due to this deletion, it didn't actually prevent the cells from making significantly more fetal hemoglobin. They also checked for other rearrangements, like inversions, and found those occurred at a very low rate.
Okay, so some on-target rearrangements, but maybe not functionally detrimental in this context. What about off-target edits? CRISPR hitting the wrong spot in the genome.
Always a major concern. They used a technique called circle seg, which is quite sensitive, to map out potential off-target sites in vitro first. Sort of flags anywhere the CRISPR might cut.
And did they find actual edits at those flagged sites in the cells?
No, they didn't. When they performed deep sequencing on the edited HSPCs, specifically looking at those candidate off-target sites identified by circle seg, they couldn't detect any mutations.
That's good.
And they also did standard checks like karyotyping and Feisch analysis on the cells after transplantation in mice. And these didn't reveal any large-scale chromosomal abnormalities either. So, within the limits of their detection methods, the off-target risk seemed low in these preclinical models.
Okay, that's reassuring. The study also mentioned something about improving the Cas9 enzyme itself, getting better results.
Yes. They compared the standard Cas9 enzyme, which usually has two nuclear localization signals, or NLSs.
Those are like the get into the nucleus tags, right?
Exactly. They compared that to a version with 3 NLS, Cas9 3X NLS, and they found that this 3X NLS version gave them more consistent and generally higher rates of editing in the HSPCs.
Interesting. So even tweaking the delivery tool can make a difference.
It suggests that optimizing every component of the system can lead to better, more reliable outcomes.
So pulling the findings from this blood advances study together, it sounds like there's pretty solid preclinical evidence for this approach, right? Boosting HbF via CRISPR editing of that BCL11A site seems, well, feasible and potentially safe in these models.
I think that's a fair assessment. The study presents a strong case. It shows you can get that HbF boost, it persists, it happens in the right cells, it reduces sickling in a disease model, and the safety profile based on their analysis looks manageable. Definitely supports moving forward.
Now let's pivot to those opinion pieces and this whole debate around the word precision. One of the main arguments seems to be, this works maybe, but it's not precisely fixing the sickle cell mutation itself.
That's the crux of it, yeah. The critics point out that targeting a regulatory element, this BCL11A site, to switch fetal hemoglobin back on is fundamentally different from correcting the single DNA letter mistake in the actual HBV gene.
They call it a workaround or even a brute force approach, not an elegant fix. Why that kind of language?
Well, the elegance, I suppose, would be that perfect single-base correction of the original mutation using something like homology-directed repair, or HDR. But HDR is known to be pretty inefficient in stem cells like HSPCs.
So it's technically very hard to do that precise fix reliably in the cells that matter.
Exactly. So the argument is that this current strategy, while clever and potentially effective, is essentially a pragmatic solution born out of the limitations of achieving that truly precise, elegant correction right now. It's seen as bypassing the problem rather than solving it at its absolute root.
And this technical reality seems to fuel some ethical concerns, particularly around how these therapies are described.
Yes, there's definitely unease expressed in those pieces about overstating the precision. While CRISPR itself targets a specific DNA sequence precisely, The outcome here isn't a precise fix of the original error. It's a broader physiological change induced by disrupting a natural regulatory pathway.
So the worry is that calling it precision medicine might be misleading.
Potentially, yeah. It might give patients or the public the impression that the underlying genetic defect has been perfectly repaired, when in fact the original mutation is still there. The therapy works by compensating, not by direct correction.
The opinion pieces really focus on the language used by the media or maybe the biotech industry, sometimes calling this a cure, without that crucial clarification.
Right. They argue that while the clinical benefits for patients could be massive, life-changing, even labeling it a cure is problematic if the root genetic cause hasn't been eliminated. They stress the importance of transparency, making it clear that this involves permanently disabling a gene's regulatory switch and relying on fetal proteins being active in adults long-term. It's about ensuring informed understanding.
So it's this distinction they draw between something being, say, scientifically impressive or technically clever, which this arguably is, and whether it's being marketed or described with complete honesty about the mechanism.
Exactly. It's not necessarily questioning the value of the therapy, questioning the accuracy of the description. Is precision editing the right term when you're knocking out a regulatory site rather than correcting the disease-causing mutation itself? They argue for more nuanced language.
Okay, let's try and connect these two threads. We have the Blood Advances study showing strong preclinical data for this HbF induction approach.
Yeah, which is really significant progress and shows the potential of this specific strategy.
And then we have this debate, highlighted by the opinion pieces, about what precision truly means in this context and how we should talk about it.
And I think both are valid points. The science shows this workaround can work, potentially very well. The debate reminds us to be careful with our terminology and not let the excitement obscure the biological details.
So it's less about, is this therapy good or bad, and more about, are we describing it accurately, considering the initial promise of CRISPR for direct gene repair?
Precisely. It reflects where the technology is now. We can target DNA very specifically, but achieving that perfect single-base correction efficiently and safely in the right cells for therapy is still a huge challenge. So we're seeing these clever indirect approaches emerge as potentially faster paths to clinical benefit.
It's interesting, too, that the Blood Advances paper itself, while championing its approach, does mention future tools like base and prime editors, right?
Yes, absolutely. They acknowledge the ongoing development. Base editors and prime editors are designed to make those more precise changes, potentially correcting the actual HBB mutation without needing the less efficient HDR pathway as much.
So the scientists are aware of the limitations and are working on the next generation.
Definitely. What we're seeing clinically now with HbF induction might be the first wave, using the most robust available tech for this application, while research simultaneously pushes towards even more precise solutions for the future.
Okay, so let's try to wrap this up. Our deep dive today suggests CRISPR can be used effectively to boost fetal hemoglobin for sickle cell and beta thalassemia by editing gene ranges.
Yeah, with promising preclinical results showing efficacy and addressing safety concerns in those models.
But critically, it's not a direct fix of the underlying mutation, leading to this important discussion about whether terms like precision or cure are fully accurate here.
Exactly. It highlights the need for really clear communication about how these therapies work, their benefits, and also their limitations compared to the ideal of perfect gene correction.
It's a powerful example of scientific progress, but also a reminder to think critically about the narrative surrounding new technologies.
Absolutely. The science is impressive, the potential impact is huge, but honest and nuanced communication remains paramount, especially when dealing with people's health and hopes.
Which leaves us with a final thought for you, the listener. As these incredible genome editing tools keep evolving, how do we balance the real, tangible benefits of these perhaps imperfect but effective workarounds that are becoming available now against the longer-term goal of achieving truly precise genetic cures?
And what frameworks, ethical and practical, should guide how we talk about these advancements and how patients make choices? And the complex intersection of cutting-edge science, communication, and societal values that we'll surely be navigating for years to come. Definitely worth exploring further.
All right, y'all. Hope y'all were able to hear all that. That was the Science News Podcast, episode 4, available now on YouTube right now. Link in the description below if you want to go check that out if you haven't already. And as you can see from what's sharing on my screen, this is in the textbooks. This textbook came out, I think, in like 2011, maybe later on. So obviously we're later down the line as far as how this was conceived conceptually and where we're at now. I could be wrong. This could be a little later. This could be like I should probably check. Let me not. So we're what page? 98. Let's go up. 2018. 2018. So it's a lot more recent. A lot more recent than I was saying. But the point being, I believe what is being discussed occurred in 2019 or 2021, around that time. And I know if you're following along on the channel, we haven't gotten to this chapter on amino acids. We're still on chapter 2, water. But for the sake of this podcast, I wanted us to jump over to where we were at 73, 75, 77, okay, 98. So we're going to go a little slower when we actually go through this and go through the textbook. But I was really interested in dropping this podcast right now because there was a video on YouTube by a creator who's pretty big, recently released a video on CRISPR and had an interview with, the Nobel Prize winner, Jennifer Doudna, and they were discussing, this breakthrough achievement in the science field of how someone has been cured of sickle cell anemia, phenotypic expression, problems, discomfort using CRISPR. So on the surface, and that's pretty much what I feel like is a big concern, is this surface level of tension. Lots of money being shifted around, lots of research, funding, and attention has been put on CRISPR. And the way it's been marketed and sold and released to the public is this narrative of a scalpel-like precision. And I thought one of the most interesting moieties or motifs that we see is this juxtaposition of discussing CRISPR, new technology, scalpel-like precision, and its ability to, cut out whatever we want in the sequence, cut and paste, right? That was the whole idea, cut and paste. And they juxtapose it with sickle cell anemia. Always. Clearly, it's in the textbook this way, right? We're talking about it. And in the video and a lot of this media attention in the science world, it demonstrates this juxtaposition of this super clean cutting, tactic or technique and sickle cell anemia. What is the big deal about sickle cell anemia? Well, as you heard in the podcast, or we can talk about it right here, if we can get to it, right here. Sickle cell anemia is called by a single point mutation. Here's normal hemoglobin, sickle cell. What do we see right here? Instead of a glutamine, we see a valine. This single change in a residue. You see that? Glutamic acid has been replaced, mutated to a valine residue. And you might be wondering, what does that do? What does that do? Well, we have to know what that amino acid looks like and what a valine looks like. And I don't want to get in trouble and get my video blocked by searching, so I honestly thought it would show it, but it's not going to show it. Basically, let's see if I can pull up the amino acid valine versus glutamic acid. They have different properties. So glutamic acid has a longer side chain with a carboxylic group, right? So it can be deprotonated and protonated acidic states. Meanwhile, we have valine, which has a branch chain structure of hydrocarbons, non-polar. Right? So we have something which is polar, and then the mutation results in a non-polarity. And anytime we see such a drastic change in electronegativity and polarity, and that will affect binding affinity. Okay, so if we jump back to here, we're affecting binding affinity. And the main thing that I was getting at was, wow, this seems so intuitive, right? We have this scalpel-like precision tool, and we know exactly where we want to cut. We only want to change one thing. We're not trying to do some crazy elaborate surgery or suturing, right? Like some systematic recombination, the true cut and paste. We're literally just trying to be like one spot. And yet that's not what we do. That's not what science is doing. They're doing something totally different, which actually resembles how we've basically annotated genes this entire time. It's a brute force method, a knockout method. And that's been the tactic this whole time. All we did was maybe change the tool from zinc finger, maybe a zinc finger nuclease or exonuclease, endonuclease, and now we're using a CRISPR-Cas9 binding. But we're not changing the one amino acid that is incorrect. Isn't that crazy? Even though we can find how that is translated in mRNA, and yet we don't go through that method and say we do something totally different. We go to the region of where that regulatory system, there's like a cluster of regulatory mechanisms at place for enzymes, the whole pathway, and then the regulatory mechanisms. And that's where that, so if we go back to it, and get back to it, see where it talks about it. That's where that region is, BCL11A. This represses the production of fetal hemoglobin. So the big thing here is that we're being told and sold of CRISPR being this super precise, incredible, breakthrough, revolutionary technique that can literally be a cut and paste, as you see on a computer. Cut and paste, cut and paste. And yet we don't use it that way. If we did, we would use it in the most simplistic way, changing one single amino acid. That's how we would do it. But we can't do it. We don't do it that way. And yet we're still talking about CRISPR in this precision type way. And that is what needs to be checked. We need to put that back in check. We need to check the marketing of how science is being sold, because it's all great if the lore sounds really good. But what we actually do, there's deception, CRISPR deception. I'm not saying there isn't, breakthrough stuff in the works, but y'all, it's not where we think it's at, where we want it to be at. And this is a workaround. As we see here, the differences between adult hemoglobin and fetal hemoglobin, we're talking differences in these chains, these residue chains, right? So hemoglobin itself, maybe we need to take a look at hemoglobin itself. And itself is, again, we haven't gone over this and we're going to go deeper into it, but it's a protein made up of multiple parts. That's what you have to realize. Here we go. It's a protein made up of 2 alpha dimers and 2 beta dimers, and they together create a tetramer. So it's a polypeptide chain tetramer, and oxygen binds within it. And we start to see these conformational changes, or changes in this binding site, this location. Okay? And when we talk about adult hemoglobin and fetal hemoglobin, we see differences in affinity for that internal binding molecule. So we haven't gone into BPG and that kind of stuff, but the main thing is that we're expressing something that is expressed as a fetus. Why would you need higher affinity as a fetus for oxygen? Because the fetus is getting oxygen from the mother. So if it doesn't have a higher affinity for oxygen than the mother, it would never be able to get some of that oxygen from the mother. The mothers would just keep cycling its own oxygen. But the fetus, the mother, is breathing for herself and another organism, the fetus. And so the fetus has to have that higher affinity. And if we're expressing this over affinity for oxygen, that's going to have problems with the reverse. There's going to be lots of problems down the line that just aren't even talked about. And in the video, it mentions some of the checks that they tried to do and talk about, but they also brushed over it. And I get that that's, the problem with summarizing research, especially through these machine learning models and AI models. And especially when there's a bias, a clear bias, for it to try to seemingly seem extra positive and not be skeptical, to the point that I'm trying to see this and demonstrate to y'all of how it should be. You should be more skeptical about this kind of stuff, because you can't just make a single cut right here. It's going to make cuts down the line over here, down here. Granted, this is the amino acid residue, and it's going to make cuts for the genes that are eventually translated into amino acids. But the point being, we're not getting precision cuts. We're not just swapping out amino acids like individually. It also, the research also shares how brutal this technique is for treating sickle cell anemia. I mean, the electroporation, chemotherapy, it's brutal. It's not a clean cut precision. It's not just, hey, inject this CRISPR and let it just go to where it needs to be, because it, has the Cas9 system. It can go right to where we want, and it'll just clip and do what we want. It doesn't work that way in reality. Theoretically, right? Conceptually, that's how we perceive it. And unfortunately, that's how it's even sold to a lot of people. Granted, I mean, there's people going through serious conditions and dealing with a lot of real pain and problems. So anything that could help with that could be revolutionary, could be changing. But don't sell it as a cure. Don't sell it as CRISPR is fixing it. We're kind of just like, it's a workaround. It's a workaround. And we're not doing it with the precision that we think we're doing or that we're being told. Granted, the effect is still the same, right? They're like, we're still producing fetal hemoglobin, which will mask the phenotypic problems, but we're literally cutting up an entire regulatory system, and it's unchecked and it's permanent at that point. That's problematic, y'all. That's uncontrolled. That's not scalpel-like precision that CRISPR is sold as. So we're going to build up the repertoire of knowledge needed, the foundational knowledge needed to understand more about proteins and enzymes and conformational changes and these components, these monomers of these polypeptides, and to get a better sense of how binding affects changes and ligands and receptors. And we're going to dive deeper and deeper into that. But the main thing with this podcast that I wanted to just like talk about, because personally, 2019, I'm pretty sure was when I stumbled upon, hearing about CRISPR, and that really got me into science again. And obviously that and COVID, it just brought it back to the forefront. And I was like, wow, this is where we're at. Cut and paste. This is where we're at in reality. And, so many years later, 6 years later, this is how we're using it. This is how we're using it. We're using it still as a workaround. We're still using principles of knockout genes. How do you want to know what a gene does? Well, we'll just knock out a piece of it until we notice something change. And then we're like, well, that did that. I kind of touched on something like that when it came to one of the earlier Science News podcasts, and I was talking about the communication and science's problem with trying to understand things. We're using these techniques that we're requiring a change of signal or change of data for us to comprehend a property of what we're examining. So if there's some communication going on, we won't know exactly what that communication is abiding by, or what the language is, without us taking something away and seeing that change, or adding it and seeing it change. And that's very primitive. You know what I mean? It's not precise. And that's been the typical way of doing something, right? If we knock out this gene, oh, now we lose this enzyme. Okay, now we know that it's mapped to that. But there could be a lot more things going on in that communication that was first like this. And every time we knock something out, we're losing a piece of that communication until we see something that's held together by a single thread. And then we knock that out. Now most scientists will see, then that final thread was the true connective principle, technique, or bond. Meanwhile, it was really all of that. And so we lose a lot of the connection. We lose a lot of the information in science when we take techniques that are brute force, that are just knock out, see the change, knock out, see the change, replace, see the change. Granted, we're getting better. Bioinformatics and these dry lab techniques and approaches are allowing us to do things structurally and predictively, computationally, and that's helping a lot. It's definitely guiding future research and current research now. But when it comes to the lore, when it comes to the marketing, when it comes to what is sold to the public for people that aren't these deep science nerds like we are here at this channel, for the people that are just kind of like, all right, where are we at science? What can we do? You'll hear CRISPR and sickle cell anemia. You'll always hear those two together, even though we're not using this amazing CRISPR technique to literally fix the single point mutation wrong with sickle cell anemia. But you'll hear it together always. You'll hear it always together. You won't hear about it changing the BCL11 regulatory system right here, represses the production of fetal hemoglobin. You won't hear that. You won't hear that. That's a problem, y'all. That's a problem. So we gotta be skeptical of science, and we gotta question it. What else we got? So I thought I had like a little statement on that, and I feel like I did. I'm trying to find it. So I hope I didn't just erase it. But I think I got my point across, y'all. I think I got my point across. Like, there's problems with this. So with typical hemoglobin, there has to be correct levels of affinity and binding in certain areas of the body where the pressures are at certain concentrations or atmospheric pressure units. There has to be an exchange with molecules. That's why you breathe and exhale. So there is supposed to be an exchange. So if we're having too much of an increase of this fetal hemoglobin that has such a strong affinity for oxygen, it could be outcompeted as well for something else. As we didn't dive into here, but CO2, depending on where you're at in the body, CO2 can be bound higher. We're going to get to it. I won't spend too much time on that, but yeah, that's the main thing in this podcast. Like, y'all, be a little more skeptical of the research. It may come across as picture perfect, sound amazing, but we're not exactly there yet. We're not exactly there yet. We're getting there. We're getting there. We're trying things, we're experimenting, but we gotta be a little skeptical. So I'm gonna go over more of this when we get there, but yeah, we're still on water, so I think that was chapter 4. We're on chapter 2. We just touched on electronegativity, so we're gonna build on the principles of enzymes, proteins. We gotta learn what they're made up of, right? Amino acids. We gotta learn our amino acids. We're gonna get there. We just learned what electronegativity was. If you're following along on our biochem series, this is the book we're talking about. We're about to get into buffers. That's what we're about to work on. And this will set the stage for some of the biotechnology techniques that we eventually will dive into pretty soon. I think after chapter 4, right in chapter 5 is when we start to go into the techniques that are used in the industry. So, and in the lab. So dissociation constants. Okay, we're building that chemistry foundation to understand the biological or biochemical processes that take place within the body. I won't go into that. I don't feel like going into that right now. I don't feel like going into chapter 2. But research proposal. That's what we're on, right? That's what we're on. So let me see what I can pull up. I have no idea why I'm literally just on Word, and I had my my research kind of like, I don't know. YouTube is weird, man. YouTube is really weird. They censor stuff. I don't know why they're censoring it. I have no idea why. But here's what I'll do. Okay. And then we'll go like this. Save. All right. So we just have a blank document. And let me share that. Get to the screen. Share screen. Window. Blank doc. Share. You guys should just be seeing a little... Come on, screen. Work with me. There we go. And yeah. There we go. That's pretty cool. I guess that has to be on the bottom though. Sorry. I'm trying to multi-screen right here. Oh, I can have 4. That's pretty freaking cool. Hopefully you guys are seeing that one. Just to double check. Yeah, you guys are seeing that one. So I don't know why. I'm gonna just start from the new. But we're gonna title this. Been working with some titles, but it's okay. Now, systems engineering of microbial species for novel and increased secondary metabolites. The focus is specifically on fungi, but we're gonna mix them. We're gonna let's see. Let's see. Systems engineering and co-culture. I don't know. Okay, one of the titles I have is Synthetic Microbial Consortia for Enhanced Fungal Metabolite Production via Metabolic Delegation and Ecological. The titles are kind of crazy. It's getting kind of crazy. I'm trying to find a better title. So, but the point is we're trying to create a system for engineering co-culture for novel and increased production of secondary metabolites, engineering co-cultivation systems, co-cultured systems, engineering co-cultivation systems for novel and increased production of secondary metabolites in fungal production of secondary metabolites. We're going to work with that title for now. Now, here's what we need to get across. So, fungi are prolific producers of pharmacologically valuable secondary metabolites, such as cordycepin, erinacines, and ganideric acids. However, in axenic or pure cultivation or culture, many biosynthetic gene clusters or clusters of genes responsible for producing the enzymes that ultimately produce the secondary metabolites of interest. And these typically remain transcriptionally silent, and the metabolite yields are often limited by these biosynthetic bottlenecks. This project proposes a rational microbial culture platform to enhance fungal bioactive compound production by leveraging metabolic delegation, precursor complementation, and ecological signaling between microorganisms. We aim to design and experimentally validate co-culture systems in which selected bacterial and fungal partners support or activate key biosynthetic steps in Cordyceps militaris, Hericium erinaceus, Ganoderma lucidum, and others. This strategy could unlock silent pathways, yield novel metabolites, and provide an ecologically grounded model for scalable natural product discovery and manufacturing. That's like the pristine way you can talk about this. But if we're going to recreate this in some different language, how would we discuss it? So let's start with bullet points. We know fungi valued for pharmacological. What are we trying to say? They're cell factories, beneficial bioactive compounds. Okay. That's why we're interested in them. Commonly cultivated for medicinal and lion's mane, reishi, turkey tail, shiitake, cordyceps, maitake. All right, so we got that. So modern cultivation techniques. Axenic produces big, nice and big fruits, but the medicinal potency is limited compared to wild mushrooms. Indoor cultivated species are subpar. Dynamics in the wild lead to the novel and increased production of secondary metabolites, SM. Dynamics in the wild lead to novel and increased production of secondary metabolites. These are competitive versus mirroring or mimicking, mimicking natural ecological dynamics and relationships via co-culture systems should result in similar teeters or even greater teeters. I can't spell. Medicinal potent compounds, mimicking natural ecological dynamics and relationships via co-culture systems in vitro, should result in similar or even greater titers of medicinal compounds, should result in the production of similar or even greater titers of medicinal compounds via co-culture systems in vitro should result in production of similar or greater titers of medicinal compounds in our fungal species. Typically increasing the production of secondary metabolites in microbial species. Typically, increasing the production is done via genetic modification and overexpression of proteins. Typically, increasing the production of secondary metabolites in microbial species is done via genetic modification overexpression. These have the potential for complications, raise conversations, and raise concerns for ethical compliance. Typically, increasing the production of secondary metabolites in microbial species is done via genetic modification and overexpression. These have the potential for unforeseen complications and raise concerns for ethical compliance and raise ethical concerns and further raise ethical concerns and escalate and raise ethical concerns for typically increasing the production of secondary metabolites in microbial species is done via genetic modification and overexpression. These have the potential for unforeseen complications and raise ethical concerns with these therapies. Okay? What we're proposing is an alternative. We are proposing a natural alternative, and this natural alternative is backed, proposing a natural alternative backed. How should I say this? Just the mechanism itself is what we see in nature. So we're comparing wild species. If we see the dynamics and relationships in the wild, we're able to mirror or mirror those in the lab, in vitro. So we're proposing a natural alternative for producing novel and increased secondary metabolites in our fungi. Okay, so how do we do this? How do we get about this? So first thing, like we mentioned up here, we are mimicking natural ecological dynamics and relationships via co-culture systems. So typically our practice in the lab we'll keep it in the context of mushroom cultivation. However, the very principle or methodology behind co-cultivation and the rationale behind how we're going about this research has implications way beyond mushroom cultivation. There's plant tissue culture. There's algal tissue culture. There's even, this has implications on even eukaryotic cell, like human animal cell cultivation. Because think about the relationships between us as animal species and our mycobiome. So there's dynamics at play in there within our own bodies. So we're going to keep it in a finite experiment research. But as we said, the implications are vast beyond, because we're talking about mushrooms here, right? But mushrooms can quickly take on talking about plants when it comes to mycorrhizal species, right? We talk about the relationships between certain mushrooms and the trees that they're with. Okay. And so the dynamics at work. This is what we're going to elaborate. And we're going to start to understand the types of relationships that go on and lead to certain activations of genes and productions of enzymes in different species. And we may be, keeping in the context of fungi, but it's a two-way street. Let me fix that. Fungi. I'm sure that was killing everyone. So we're going to keep it in the context of fungi. Okay. So we're proposing a natural alternative for producing novel and increase by mirroring. Okay. So how to go about it? So we're going to start with what mushrooms we're interested in. Okay. And this could be, for whoever's watching, this could be any type of mushroom. You know what I mean? I don't know what you're cultivating. I personally cultivate gourmet medicinal mushrooms. I live in California, so magic mushrooms are not legal. So I am currently not cultivating those, but who knows what may happen in the future. But maybe you are cultivating those types of species. This has implications for those as well. So again, I'm keeping it in the context of things that may be related to me, but it's broader. It has broader implications. Eventually I want us to have larger tissue culture containers, right? With rooting plant tissue culture roots. And we're going to have that with bacterial species and fungal species. And we can create a whole consortia, a whole dynamic relationship on the plate or in a 3D broth. So the research that we see currently deals with molds and yeasts, things that aren't producing fruity bodies. But as someone who's a part of the commercial realm as well, I understand that we typically work with these fungi. Okay. So how are we going to go about this? We're going to choose a mushroom. We're going to go find it in nature, right? Why do we have to start in nature? It's because if you just go and get a culture online, it's going to be an exenic culture. It's going to be a pure culture, just that species. So you don't exactly have a starting point of how to mix and match or what you're going to co-culture that system with. You can do it randomly. You know what I mean? You can just wipe some yeast off on you and get some things. That's like random. You know what I mean? And we're using science. You don't need to be random, especially if you want to apply, like track what you're doing. So we're going to start in nature, and we're going to start with a mushroom of choice. Find it in nature. And when you're out there, you're going to harvest not only that mushroom, right? Normally you just harvest it wherever you're doing it. You take a sample and you'd probably tissue culture it, put it on a plate. But we're going to also do harvest, but also take samples of soil and / or bark of on or in which you found that mushroom. Okay? And what are you going to do with this? You're going to start with the mushroom of choice, find it in nature, harvest it, but also take samples of soil and or bark on it in which you found that mushroom. You will isolate all the species, all the microbial found. This will provide the starting point for what species or groups of species should be trialed for cultivation. Start with the mushroom of choice, find it in nature, harvest, but also take samples of soil or bark on or in which you found that mushroom. You isolate all the microbial species found. This will provide the starting point for what species or groups of species should be trialed for co-cultivation. We will then explore the metabolic pathways inherent of these species, these isolated species. Okay? So we're exploring the metabolic pathways inherent of these isolated species. Understanding what biosynthetic these organisms utilize and the compounds they produce, and understanding what biosynthetic pathways and compounds these utilize, and understanding what biosynthetic pathways and compounds these organisms utilize and produce will provide logical evidence for how co-cultured dynamics may play out. Okay? All right, y'all. So we're engineering co-cultivation systems for novel and increased fungal production of secondary metabolites. We understand that fungi are these cell factories for producing beneficial bioactive compounds. Commonly cultivated species that are cultivated for their medicinal properties typically are lion's mane, reishi, turkey tail, shiitake, cordyceps, maitake, and others. Modern cultivation techniques are done exenically or monoculturally by themselves. This is done for reasons of only wanting to grow what you want, that single mushroom, and you result in really nice fruits. These produce nice and big fruits, but the medicinal potency is limited or capped. Compared to wild mushrooms, indoor cultivation species are subpar. And that's because certain gene clusters responsible for bioactive compounds are not always activated or stimulated, especially when these isolated species are forced to do everything themselves. That's another. They're responsible, so all the metabolic processes from, immune protection and immune defense, along with digestion and these other pathways, these all are more critical to reproduction as opposed to some of these other secondary metabolites that may be defensive or just surplus, excess in certain cases. In nature, you don't have these metabolic tasks. Let's see. In nature, metabolic workload is divided amongst various species. Precursors and products for one species is a precursor or a product for another. Molecules, ions, and nutrients are recycled, reconstituted, and made more available when molecules, ions, and nutrients are recycled, reconstituted, and made more available when a variety of species, when a variety of organisms and their inherent metabolic pathways, when a variety of organisms and their metabolic and inherent so molecules, ions, and nutrients are recycled and reconstituted and made more available when a variety of organisms and their inherent metabolic pathways are in a dynamic relationship with each other. Dynamics in the wild leave it a not. Yep, mimicking natural. Cool. I like this. Proposing a natural alternative. Start with mushrooms of choice, find nature, take samples of soil, isolate. This will provide a starting point for what species or groups of species should be trialed for co-cultivation. Explore the metabolic pathways inherent in these isolated species. Understanding what biosynthetic pathways, enzymes, and compounds these organisms utilize and produce will provide a logical evidence for how culture dynamics may play out. I like this a lot. It's in the water. We can't. Cool. So we got that going. So now how are we going to test this? How to test for secondary metabolites? Okay, we're going to be doing things like LCMS. We're going to be doing things like NMR. We're going to be doing things like bioinformatics and cheminformatics. And then we're going to also be doing things like that. So how to test for secondary test, LCMS/GC. I guess we'll go like that. LC/GC for gas chromatography, MS, NMR, bioinformatics, cheminformatics. What else we have? We have, I forgot what I was going to say. Dang. Well, so we'll go for that. Let's go for that. Multi-omics. This is like proteomics, transcriptomics, metabolomics, genomics. Okay, so I can't spell. All right, this is good. This is a solid outline. If we break this down, how to test for secondary metabolites. So when talking about LCGCMS, what is that going to do for us? So liquid chromatography, gas chromatography, these are ways of purifying samples, of isolating, separating samples. Okay, so we're going to use various inherent properties of our molecules or samples that we're testing for, and based on those properties, we'll be able to pass them through various columns and material to separate them out and have them elute different orders. And those orders will be separated in what is called fractions. And those fractions will then be sent into another device that's usually done in tandem called mass spectrometry. And this is going to utilize inherent molecular properties of magnetism and bioelectricity, electromagnetism, and use that as a way to basically identify atomic fingerprints or molecular fingerprints. Okay, and so these molecules, based on their mass to charge, will move within our detector or mass spectrometer in a predictable way that separates them by their mass to charge. And we'll be able to map those charge peaks to specific molecules. And this separation, along with this mass to charge ion fingerprint mapping, allows you to identify exactly what's in your sample. So other things that you might do to identify what's in your sample are not going to be as specific as what you would is what you would understand or get from LC-GC-MS. So what am I talking about? I'm talking about like colorimetric assays. These are going to be the experiments and testing that is going to be quick, affordable, and very visible, right? And those will utilize principles of absorption and what's called Beer's law, and this connection between concentration or light absorption to concentration. And that will kind of limit what you can understand from it. It won't be as specific, but it will allow you to perhaps test for certain classes of compounds. And so that's what makes this more practical and affordable. It also doesn't require such crazy equipment. You can use different wavelengths of your spectrometers to extrapolate or to verify these different concentrations or classes of compounds. But again, if you want specific names like corticipin, if you want your furicinones or psilocybin, you're going to need to detect those using LC or GCMS. Now, NMR, say you have something, you got this molecule and you don't know what it is, you plug it into a database and it's not really giving you anything, you would run further tests called NMR, nuclear magnetic resonance, and you'd use this to specifically identify what the molecule you have on a much higher level. So LC and GCMS, that does rely on having databases of which those compound peaks match to, and so that's why NMR is more used for like true unknown. And there's hydrogen and carbon based for that. Now, bioinformatics, cheminformatics, that's going to build on the databases of which those techniques like LC and GCMS and NMR produce. So you're going to get data. People are going to be testing these compounds for years. They've been doing that, and they've been putting them into these databases. And these databases allow for querying using computational algorithms and techniques to basically just like, it helps everyone in their research. So you'll know things are mapped to certain molecules, organisms, and you can utilize these platforms and databases in pipelines. And so you can run your experiments of testing and isolation and then funnel that into your bioinformatics and cheminformatics computational pipeline part. And that will extrapolate other information that relates to these multi-omics approaches. These are whole areas of or domains that people focus on for medicine and for science. And so by combining all of these different things, we're going to be able to utilize or identify and test and see the differences and changes in our secondary metabolites as we go through this process of co-cultivation. Okay, so this is what we were able to quickly transpose. I don't want to get blocked for showing some super nice research proposal. So we did it on site right hand. And this is just a little quick walkthrough. I can make it nice and neater later, but yeah. So just a quick run through. What we were trying to do is engineer a co-cultivation system for novel and increased fungal production of secondary metabolites. And we're trying to do this without genetic modification. We're trying to do this naturally, right? Because we grow things organically, non-GMO. We know that fungi are these cell factories for producing these beneficial bioactive compounds. People have been utilizing mushrooms for centuries and across cultures. And commonly cultivated species are not limited to these, but in the context of what we're going to keep it in this context, in this conversation, we are going to deal with some of these strains: lion's mane reishi, turkey tail shiitake, cordyceps maitake. You know about these. You may not be able to get them exactly where you're at, but I know you've heard of them, or at least you should. And so, like we were saying, modern cultivation techniques, when you grow these in vitro and you're not just foraging, you are going to do these exenically or in a monoculture. You're going to isolate species. You're either going to buy it from someone, or you're going to take a sample from the wild, or you're going to collect spores and germinate them and infuse them. But the result is usually just an isolated culture, a monoculture, not a monokaryote, but a monoculture. Now, this isn't bad necessarily. It's good to know what you have and what you're working with. And especially in the context of commercial production, you want to produce only what you want to produce. And you want it to be predictable, at scale, and clean. So there's reasons for why we use exenic or monocultivation. However, that's for commercial yield purposes. In the context of biopharmaceuticals, biotechnology, it's not always about yield, or at least the food aspect, right? So in another context, the focus is on potency, production of novel compounds, using biosynthetic pathways to express unique, novel, or isomers of beneficial bioactive compounds. So again, modern cultivation, you're going to get these nice big fruits, but you're going to be capped, limited with the medicinal potency. Why? Why would you be limited? Why would you be able to see Why do we see differences in wild mushrooms versus indoor cultivated mushrooms? This is because the metabolic workload is delegated to our monoculture. What do I mean by that? I mean that our monoculture, although capable of it and although can produce some really nice fruits, it has to do everything itself. That's what I'm trying to say. And what do I mean by that? I'm talking about digestion. It has to eat. I can't spell. My goodness. Digestion. It has to eat. It has to grow, right? It has to defend itself and it has to reproduce. Look at all that it has to do. And all using the finite and limited space and resources provided by the cultivator. Yikes. I'm so sorry. Cultivator. Okay. Does that make sense? So yes, we can get these nice big fruits, but that's because you've given them everything to do just that. You've given them a nice comfy couch to sit on their butt and watch some TV and, get fat. It's easy. That's nice. But if you want them to be active and be this super food that you're expecting them to be, and a lot of people are quoting them to be, I'm not saying they're not healthy. They are, of course. But dude, there's a reason you're not seeing these natural products in biopharma, in a hospital. They're used as adjuvants. They're used in tandem, like together, because the potency it's that much. So you have to take it a long time, different ways, right? Over time. Meanwhile, when you're in the hospital, you need something right away. You're dealing with some ulcer or something, some infection. You need that stuff right away. So this is why you don't always see these natural products. That plus money, right? That's obvious. That's plus money. But you can produce it and sell it, and that's money. But we're not seeing the potency. We're not seeing the potency that we need. But we can tap into that. We can tap into that if we look into nature. If we look into nature, we see that they have extensive networks going on. Their resources, their communication is vast. You know what I'm saying? When you're cultivating in vitro, you are limited to the nutrients on your plate, right? So here I have an isolate. I took this from the wild, and it's kind of split because I didn't seal it and it kind of dehydrated, and that's kind of what happened. But this was a isolate from my wild oyster strain. You can see the videos on my channel, me finding it in SoCal and then taking spore prints. And then also you can see me inoculating it in a sab. So I got it on plate, then the LC, and I just put it into grain. So hopefully I'll get those all the way to fruit. But look, it starts on something like this, a tissue sample. These are samples I took a clean sample from the mushroom itself and put it on a plate. Or you can take spores. You can take spores and take their spores and then put them on a plate and have them fuse, okay? But after that, they'd go do something like this. Some people call them like bioreactors, or really it's just a container, a liquid culture container. Also, it's closed though. It's closed, right? Each cycle, it's in a closed container. It's isolated. So whatever you have or whatever you're using is limited to those resources, what's in there. Again, that's not bad, but you're limited. You're capped. So if you want to produce something, and to make that compound, there's a bottleneck upstream. You need certain nutrients or ions, right? You may not have enough to make that final product, at least more of that. And so in nature, your fungal species is going to be interacting with maybe bacteria, other fungal species, and they're going to be tapped into their own networks and their own communication, their own vat of resources. And one organism's precursor or product is another organism's precursor and product. And so things are being recycled, reconstituted, and the communication is faster and able to happen more fluently. You know what I mean? You have different structures for your fungal species to hide within, maybe plant roots. And so you get protection. And so you're not dealing with all these different tasks, this metabolic workload that we just talked about. You're not dealing with that with one species. It's given to others. And this very core principle, seen in the wild, not done in vitro, is how we're going to approach this increasing potency. Okay? And we're not doing anything too crazy, because in vitro cultivation itself took what is done in the wild and tried to mimic it indoors. But for all the reasons you can probably assume, money, time, ease, people stopped with just one culture. They're like, look at all this food I can produce, and I'm still getting good science, good research. But that doesn't mean we can't elevate our research, our practices, and the whole industry itself, especially when it comes to medicinal potency and therapy, because that's why I got into this. Food, medicine, and money. You know what I mean? That's why we're here. And we're not going to just willy-nilly throw different species at this. We're doing this with scientific precision and rational logic. So we're going to continue to look at nature and what nature does, what nature has already shown us, the organisms at play in their own relationships. And we're going to try to continue to mirror that in the lab, in vitro. All right, right here, like we said, precursors and products for one species is a precursor and product for another. Molecules, ions, nutrients, these are recycled, reconstituted. Okay, it's very, very important, very, very important. And so I get it. We got some species. Now, what do we need to understand about those species? We took samples from the wild of around where the mushroom or whatever our species organism of choice is, and we understand what organisms are there. Right? We haven't identified what they're all doing, but we have an understanding of who's there, who's going to be talking. Now we can go deeper, exploring those individual species to explore how the conversation is playing out. What are those relationships? Are they competitive or non-competitive? You see what I'm saying? Competitive, you're going to see that release of those secretion of chemicals, right? For defense, you're going to get those antimicrobial chemicals. That's good. You want those, right? So that's a way to increase production of those. Depending on which species you have co-cultured with, it's going to activate those chemicals. Maybe you have a bacteria. It's going to produce some antibacterials, antifungal species. So we can think of it in that competitive way. Now, non-competitive, this is where it gets really interesting. This is where it gets complex, nuanced, but fun, right? This is where art comes in. This is where all those different interdisciplines that we were talking about, especially computational techniques, come into play, because these multi-omics methodologies really allow us to understand biosynthetic pathways that are going on within each organism. And when you understand those biosynthetic pathways, you understand what those organisms require and produce. And when you understand that, you can understand more of the pool of resources that are available for your fungal species and what those resources can eventually amount to. So if I have an increased precursor of these triterpenoid backbones, I know eventually I can get to those final triterpenoids or those sesquiterpenes that we want. Okay, so this is how we know we can start to play with the production of what molecules we want. And when we understand those pathways and those enzymes that certain bacteria and other fungal or plants utilize, we'll be able to integrate that or incorporate that to supply that metabolic task or work or network resource for our fungal species. Okay, hopefully I wasn't just beating that into a pulp, but I hope that made sense. So there are these dynamics in the wild that lead to wild mushrooms having such potent compounds in them. And it's based on, or it's triggered by the competitive and non-competitive interactions and relationships that they are exposed to in nature. They're exposed to trike, but if you try to expose trike to your species in the vitro, trike's going to win. So you're missing something, right? So you're missing something in the dynamics. That two binary dynamic doesn't work. You need some biocontrol agents. You need some other organisms that can keep trike in check, protect your fungal species. So there has to be these balances, these checks and balances that are at work. And when you're dealing with co-cultivation, maybe you're not seeing what you expected or wanted. Maybe that's because you need another organism and its inherent pathways, molecules, and properties to balance out and keep that trinity, or not even three-way, that dynamic relationship in check and in order, balanced. So we're going to mimic natural ecological dynamics and relationships via our co-culture systems. So we're going to take our isolated species and then we're going to cultivate them. Imagine if these are the same species, but imagine if these were different species. You see what I'm saying? So we're going to put them together and they're going to interact. If you've ever bred mushrooms, you'll sometimes see zones of inhibition, right? These areas of interaction that show like, oh, they can interact or no, they can't. And sometimes metabolites are produced in those divisions and those points of interaction. It's hard to show you guys right here. So when you start to co-culture, again, this is the same species, but imagine them different. They're going to activate different genes and production of enzymes in those species. Now, you can either, what the research shows, they'll take mono or isolated cultures and they'll either plate them together, or they'll take isolated liquid cultures and then they'll combine the liquid cultures and have them grow out. Now, a problem that we see is that the growth is different. one strain, as you probably know, one strain grows at different speed or is able to break down the nutrients faster. And so you get this non-consistent or inconsistent data when in your trials, in each trial, right? If I put the same size wedge, the homogeny of your nutrients may not be the same, or the temperature, what's exposed, the light, all these different factors can deregulate or destabilize the controlled growth that you thought you would have, leading to inconsistencies in each trial. Regardless, they grow. They grow. And when they take the supernatant or the excretion of the fluid of which they're in, as well as lysing open the cells and extracting that, combining the fractions, and then or continuing to isolate them, and they test those isolates, they're able to see increased and novel compounds produced. That's incredible. That is incredible. And look at the stage that it's done at. It's done not at the fruiting body stage, at which common cultivation is done. They stopped right here at LC. That's the second stage. They didn't even go to grain or substrate. Now, granted, I have problems with that because based on nutrient exposure, that's going to activate the production of certain genes and enzymes and pathways, right? So that's why there needs to be research at every stage. However, the foundational knowledge and the testing of this logic, rationale, methodology is there, and it's vetted. So we got to expand on it by using different species, right? Because they're usually doing like Saccharomyces cerevisiae and non-fruiting body species. So if we want to evolve this for us, for our purposes, we're going to use our strains, but we're going to use those same techniques, and we're going to need to combine our research and work together because, man, as you saw, some of the techniques that we're going to need to use to test this stuff, LC, GCMS, NMR, that is expensive equipment. That is expensive equipment. You know, if we want to do this research for real, we're going to need our own equipment. We may be able to get away with paying for this for some of our samples, but in the long run, we need our own equipment. Especially if we're seeing novel compounds, you're going to need to characterize those novel compounds using NMR and stuff like this. So it's very important that we understand that, look, the technique to do it is there. It's easy. And we won't always have to test every batch. Maybe we get to a point where, all right, if you add this species with this species at this specific stage in the cultivation process, it leads to the production of these classes of compounds. So then it just becomes your standard process, just like it's your standard process to go to grain LC or plate LC grain substrate. Maybe the process evolves from plate to co-cultured plate to re-isolated, you know what I mean, or to LC and then re-isolate it. So we'll get to that point where we understand it more, and then maybe it'll just become a standardized practice to introduce this species with it, and that final product almost always has a higher titer of medicinal potency. That's the goal. That's where we want to get to. We want to get to that standardized final goal. That'll be nice because that'll be a way for us to introduce this practice into the commercial realm where they don't really have the time or desire or even a real strong immediate need to add to their practice when they're already making money. They're already doing it with just that, and they know what they're doing already. So if we want it to evolve and elevate the entire industry, we're going to have to standardize the practice. And for us to do that, we're going to have to do this foundational experimentation and research. And we're laying out the rationale and foundation to approach this research. Now let's combine as a team and unite and do this together, y'all. Okay. So again, the methodologies will be simple. We can come together and send off our samples or come together and get this equipment, but you can integrate these co-cultivation practices now. Okay. You can. And that's how we're going to go about it, y'all. I love it. So that brings me to us trying to build the research team. You know what I mean? I'm trying to build a research team, y'all. And if you're interested, reach out. Reach out on Instagram, Lab Coat Mushrooms, subscribe and follow. But let's get the communication going, you know what I mean? Or maybe you just like what I'm doing and like what I'm about and what I'm bringing. You can support my research. You can support this research still, even if you're not going to be doing it yourself. I've got a Venmo link in the description. When I do these live streams, you can donate or whatever. The funding is going to this research. You know what I mean? I'm locked in. I've dedicated my life to the sciences and to health and wellness. So I'm growing this food and doing this research for y'all. You know what I mean? Obviously, I'm gonna take it myself, but I don't just do this or talk about this for myself. It's for everyone. You know what I mean? And that's why I'm sharing this stuff with you guys. We're gonna build this research team, and it's gonna grow. It's gonna elevate everyone. So let me I was just going to save this, but I'll just switch screens. So yeah, y'all, what do y'all think? What do y'all think? Stop sharing. Stop sharing. What do you think? What do you think of this research? Cold cultivation. Does that make sense? Makes sense, huh? And it's already happening too. And they're not doing it with our same intentions as far as with our strain choices. But yeah, what do y'all think? You see, you want to see the grant? So, this type of research is pretty big. You know what I mean? So obviously there's stuff that we can do here at home, but I'm trying to formalize this, y'all. I would love to present this research and build a team of researchers and go about this the formal way. Be back, y'all. Sorry about that. All right, peeps. I think we're going to end the stream there. That was good stuff, y'all.